Students, go ahead and title your notes Gene Regulation. All right, we're going to get started here. So just a few bullet points, a couple of things that, I don't know, maybe may or may not need to be said, but maybe you didn't know this or maybe you didn't think of it this way. But every cell in your body has the same genes. However, cell types, I guess you could say, vary dramatically. So, for example, you could have like if you looked at a brain cell, which would basically be a neuron, and you compared that to say a muscle cell, they would be so dramatically different that you could hardly even begin to compare them. But you know what would be amazing is that if you looked at the DNA inside, it would be exactly the same. So how then? So the question then is, how then can you get such differences in these cells if they have the exact same DNA? And the answer is gene regulation, right? So in other words, some genes are on/off let's just say genes are on and off, and there's variability there. So, for example, brain cells have the insulin gene, but it's inactivated. It's not, in other words, it's not expressed. So that's just one example. Whereas a pancreas cell, insulin gene is expressed. Okay, so that's just one example, but there's many other genes we could talk about that would be expressed in one cell type and not in another. So there's a couple of places where we can regulate whether a gene is expressed or not, right? And it could be here in transcription. So we could affect things here. In other words, we could either inhibit transcription or we could try to initiate transcription. And that would have an effect on how much protein is made, right? Because then you got your mRNA, and then down here you'd have the final protein. So if we inhibit transcription, if we completely inhibit transcription, we will eliminate this protein. It just won't get made at all. So we're going to focus today on this, where I put this star, and that is different ways in which prokaryotes, specifically we're going to focus on prokaryotes today, how they go about regulating genes at the level of transcription. So add a new title that says gene regulation in prokaryotes. Okay, so remember what a prokaryote is? It is basically an archaea or a bacteria. So it's a little single-celled organism, very simple, primitive life form compared to eukaryotes. And so they actually have a different method of gene regulation. And they utilize something called an operon, which is a gene switch that blocks or induces transcription of a particular gene. So again, I told you we'd be focusing on the mechanisms that work at the level of transcription. So that means then that we're going to be looking at RNA and the molecules that interact with an RNA strand. So go ahead and actually that's not true. We're going to be looking at DNA. I shouldn't have said it that way. We're going to be looking at DNA, and we're going to be looking at the molecules that interact with the DNA to either prevent transcription or initiate transcription. So if you could just draw a big long piece of DNA, and remember that DNA is like a double helix, right? So these are the nucleotides, the rungs of the ladder. And then over here, this little segment right here is called a regulatory gene. And it, when it is transcribed and translated, it produces, right? That little gene will produce a messenger RNA. And then when that RNA gets translated, it becomes what we call a repressor protein. And then the repressor protein comes back around and influences the target gene. So down here, we're going to go ahead and kind of just shade in this area here because this is the target gene. Or I guess you could say the gene being regulated. Put that in parentheses, the gene being regulated. Fill in the blank. Could be hemoglobin, it could be whatever. But anyway, and we call that downstream. So the reason it's downstream is because transcription is going to take place in this direction from left to right. And so anything that we do up here is going to influence what happens downstream. So I told you that this repressor protein is going to come over here and interact. So there's this little spot right here called the operator region. And that's where the repressor will bind. So there it is. The repressor came in and binded to the operator region. And the effect of that is that it blocks transcription. Because the promoter region right here, this is called the promoter region. This is where the RNA polymerase would normally bind, right? So you got RNA polymerase. The RNA polymerase would like to proceed and move from left to right and initiate transcription, but it now has an obstacle, right? The obstacle being it's physically blocked by a repressor protein. And so it just cannot proceed. And so in this particular case, the gene is considered off. The gene is off. It is not expressed. Why? Because the reason why is because we have the repressor protein actively blocking the blocking transcription. Okay, however, I do want to point out that there is another possible interaction here, and that is that there are other molecules called inducers. And what they do is they bind to and inhibit the repressor. Okay, so students, this is a double negative, okay? Everybody see why it's a double negative? What do you get if you inhibit an inhibitor or you inhibit a repressor? Then it becomes positive. So the inducer actually has the effect of promoting gene expression. Now, why is that? Because an inducer inhibits the inhibitor. And how does it do that? The inducer will bind right here to the repressor, and then it's going to prevent the repressor from binding up here to the. In other words, if the inducer binds to the repressor comes off and polymerase can move forward. So we can say that. We can say, let's add that right here. If it binds to the repressor will be removed. It'll just kind of pop off of the gene, and then transcription will proceed as usual, proceed normally. Okay, so let's just define a few things here. I want to get just bullet point definitions. The operator, what does that mean? It is the site where the repressor binds. What is the promoter? The promoter is the site where the RNA polymerase binds. What is the regulatory gene? Well, this would be the gene that produces the repressor. What is an inducer? We've kind of already stated all of this in the notes, but I'm just giving this to you in case you forgot. A molecule that binds to and inhibits the repressor. All right, good. So that's the summary of it all. Any questions about it? Yeah. Can I what? Oh, yeah, sorry. There you go. You betcha. All right, so today in the PhET simulator, you're going to look at a specific operon called the lac operon. Do you guys remember when we made yogurt in here? Yeah, that was cool, right? So when those bacteria, when they got immersed into the lactose, and lactose is milk sugar, right? As soon as those bacteria were placed into the lactose environment, the lactose permeated through the cell wall. It got into the cell, and those bacteria, the lac operon, got activated, and lactase started being made by those bacteria. And what actually stimulated the production of lactase was the lactose, right? The lactose molecule binds to and releases the repressor, and then lactase gets made. And so those bacteria that make the yogurt, they're able to respond to their environment. When they find lactose in the environment, it activates the enzyme, the gene that produces the enzyme to digest the lactose. So you're going to look at that. You're going to look at exactly that example today. And you have some questions to answer. But we'll go ahead and end the notes here.